All right, fantastic. So welcome everyone. We are just about to start a session for the Research Data Alliance. Let me see if I can move the slides a bit. And to start with, let's acknowledge the land that we're standing on and its original inhabitants. The session is titled Linking Pixels, Proteins and Populations. And it alludes to pixels being images, and these pixels can represent any number of things when we are in the life sciences. So you can have a very complicated and interesting phenomena captured on images and being hidden that you can explore to a very high degree. You have proteins and that's being linked to different genomic and sorry, can we turn off the music? All right. And then we have proteins which are linked to different products in the life cycle of the body and the organism. And there are inherent links between these products and technologies that allows you to study any one of these dimensions. And then this is usually connected to populations of different types of organisms. And it could be populations of people during certain events. It could be populations of animals or plants or whatever. So here we're looking at integrating data across life science domains or life in general. And we will have some representation from people working on infrastructure and also building portals and support for being able to integrate and work with these kinds of resources. This session is organized by an interest group in the Research Data Alliance. So if you're a newcomer, interest groups are these platforms for bringing in people to have discussions over long periods of times until the kind of core of the interest group turns outdated or the interest is ebbing out. So we've been around for quite some time. We started in 2022 with some partners from Australia, from the United States, and from Africa, and from Europe. We are actively looking for representation in other regions as well. So if you are one of those potential representatives or know of someone, please do let us know. And there's a website that you can go to and explore. So the Research Data Alliance website, it's a free membership, you can register. And you can join our interest group if you want to follow along activities at future plenaries. For this session, we're going to use something called Mentimeter for the Q&A and also for a very quick check-in. So please scan this code. It's going to stay up here on the screen for quite a few slides, and I'm going to pop it back just before we jump into the Mentimeter responses as well. There's also a collaborative notes document. You should be able to reach that through the online platform. And if you have a computer, I would highly recommend you to open this document so you can check in and add your name if you're interested in following up. And at the end, I will also prompt you to maybe add some information about your group or your organization if you would like us to kind of continue this discussion and invite you or acknowledge some of the work that you've done in those discussions. So there's a short code, wna.se/multi-modal. And if you're on a phone, you will also get the link through the Mentimeter. All right, so we have today's meeting. The meeting agenda you could have looked at that before the meeting. But in general, we're talking about multimodal life science data. And by multimodal, we basically means different types of data, or it could be different types of experiments that look at the same problem. And we're going to have a few talks that illustrate this with examples. And we're going to have a discussion on, what do we actually mean by multimodal? Because I have experienced that talking to different people will be thinking about different types of research studies or different types of requirements. So this is a first exploration with the speakers and also you here in the room to try to detangle and finding kind of a starting point for some continued discussions, and maybe also finding some relevant standards that we can start working from. And the agenda is a 10-minute introduction. I'm going to show a few Mentimeter questions as well. This is basically where my presentation and introduction stops, and we're going to have 4 speakers entering the stage, and I'm going to introduce them as they appear. But basically setting the scenes here, and each of the people stepping up are representatives of different types of infrastructures. We're going to have a Q&A, and you can ask questions through the Mentimeter. There's a button at the bottom where you can open the Q&A. You can also vote for others' questions. So please do add some questions as the speakers are talking and when they arise. And then we will address all of the questions at the same time after the presentations have finished. After addressing the Q&A for the presentations, we're going to have a discussion more openly with the audience and see if we could agree on some potential next steps, and if there is some sort of state-of-the-art that we can summarize based on the discussions today. So the end results are going to be captured in the collaborative notes document. So we probably won't be able to populate it to a great extent today, but I'm going to go back to it and fill it out once the recording is available. And I hope it can be a useful document then to spur some discussions down the line. So the document should look something like this, and if you jump into it, you can add your name to start right away. So I'm going to switch over to the Mentimeter. There we go. There we go. And the first warm-up question here is basically, so where are you from? It's nice to see what kind of distribution of people we have here today. The speakers have a kind of high weight towards the European area, but I'm hoping that the discussion can address that to some degree. So I'm going to leave this open for a few more seconds. So a lot of Germans. Good to see that people have taken the time to travel all the way over here. So we have someone from Japan, Tasmania, Australia, and Australia. I'm guessing that's a region, not being really familiar with Australia. It's a state, fantastic. And we have Czech Republic, United Kingdom, Greece. All right, fantastic. So I'm going to move on to the next one, which is a little indication of why you're here. And there are 4 options. I mean, if you don't feel this fit, try to select one anyways. But I mean, why did you intend to join this session? Do you have something that you want to bring with you and share with the others? Or are you looking for some collaborators, or just interested and curious about the topic in general terms? So I see there's a lot of people who are here to learn. Some of you are here to share. And I'm very happy to see that some are also here looking for collaborators. So I think that's one of the end results that I'd really like to see after this session, people getting together and really trying something to work on together. All right, so I'm going to move to the next slide after this. And this is basically a direction to go into the collaborative document and add your contact information. And as you do that, I'm going to bring up the first speaker, which is Tony Burdett, and he's going to talk about biofair from the UK.
Okay, thanks, Wolmar. Oops, sorry. Okay, great. Yeah, so today I'm going to talk a little bit about some of the use cases that we have for life sciences multimodal data integration. That's this one here. So yeah, thank you for joining. So first I thought I should open with a slide about what BioFair actually is. We're a relatively new federated digital research infrastructure for the UK, targeting a sort of step change in, in fair data practices, fair research data management practices for the UK. So we're a new digital infrastructure. Fair is in our name. We're obviously sort of focused on trying to create that culture change around good data management practice. But we're also sort of thinking a bit more holistically than that as well. So we're thinking about how to bridge researchers into research data management, into service delivery providers. But culture change is kind of the theme for this presentation this morning. Just to give you a bit of a background into BioFair, we've got sort of 5 headline objectives. Culture change I've already talked about. We're thinking very hard about defragmenting the service space and increasing the sort of coordination of the landscape within the UK. We're thinking about democratizing data access, attracting, retaining and developing research data management skills, and sort of our ultimate aim is to drive the community towards greater data reuse. That's a very quick introduction to what BioFair is. I wanted to give you, because we're relatively new, I started in post in March this year, and we're sort of about 1 year into our program at the minute. I wanted to give you a little bit of a view of some of the landscape scanning activities that we've been doing within BioFair, and how I sort of thinking about multimodal data integration as one of the threads of that. So I sort of came up with, over the last sort of few days, I came up with these kind of 3 conceptual layers, which sort of I will sort of explain a little bit and then go through some very quick examples. So broadly speaking, and we can talk about the terms, I've been thinking a little bit about assay-level multimodal data integration, which I'm sort of defining as one experiment, but with several different techniques or data modalities that generate data in the context of a single experiment. A sort of experimental layer of multimodal data, so sort of several coordinated experiments that are probing the same ultimate phenomenon somehow, and then cross-domain data analysis, which is really kind of widespread reuse of data from multiple different domains to potentially do some exploration or hypothesis testing at a very, very broad level. And I'll give you some examples of some of the initiatives that I've been personally involved in or been speaking to collaborators about over the last sort of couple of years, really. This goes both into my time as director of BioFair and also from beforehand. So just to give you some examples of a kind of multimodal assay approach. So this is, again, integrating data from several different assays within a single experiment, trying to get a high-resolution picture of biological function. And just to give you some examples, the work that I'm sort of talking about here is loads of people's work. There's way too many people to acknowledge, so I've tried to include all the links on all the slides to the respective papers. But I picked out 2 examples here. One is the sort of Web Atlas initiative from the context of the Human Cell Atlas. And in this scenario, you've got it's spatial transcriptomics and single-cell RNA sequencing data that has been sort of overlaid and resolved at a cell location level. So combining both the strengths of sort of Visium imaging techniques and then single-cell transcriptomics, single-cell RNA sequencing, and sort of deconvoluting the results so that as you probe through the sort of visualizations here, you can look at spatially resolved cell types and then examine the kind of the gene expression profiles of those from the RNA sequencing methods. And likewise, on the right-hand side, the left-hand side's an example from the Human Cell Atlas. On the right-hand side, there's an example of doing this in wheat from the Elam Institute in Norwich, where you've got this kind of expression profiles within plant cells, within wheat cells, showing the levels of expression spatially resolved into the sort of spikes of the wheat, which I think creates these kind of beautiful images. So this is kind of that first example of where you're essentially doing a single experiment. You're trying to spatially resolve it, sort of cell types and gene expression profiles of those cells, but doing it with quite tightly aligned and integrated technology in the context of a single experiment. In the second context, we've got this kind of multimodal experimental approach, and here I picked out an example from the Morphic Consortium, which is a project I was previously involved in before I started at BioFair. which is a big NIH-funded initiative to probe the function of all human genes. And here you can see on the left-hand side quite a comprehensive overview of the diversity of experimental techniques that are being exploited in the Morphic program. So broadly speaking, there's molecular phenotyping and cellular phenotyping experiments that are being done. You've got single cell gene expression, metabolomics and lipidomics at the molecular level, and then also things like pluripotency analysis and sort of embryonic organoid kind of formation from induced pluripotent stem cells. And you can see some sort of early results popping out in the visualizations at the bottom right here are early results on the Morphic portal that you can go play with. So here, this is very much about a sort of aligned experimental approach. There's sort of multiple centers generating data from the same cell lines, doing multiple different experiments, different readout types of assay. But they're all ultimately trying to shed light on this kind of phenomenon of if you knock out a gene in a human cell line, what happens to the resulting phenotype, and sort of compiling that data into gene-level summary information. The third example here then is this kind of cross-domain approach, and I picked out a couple of examples here, one from the Hollow Foods project and one from a digital twinning project that Rothamsted Research are running. And this is a scenario where you essentially take multiple different modalities of data. So the Hollow Foods project, for example, has looked at gut microbiome of farmed animals, chickens and salmons and stuff like that, and then also looked at both the genomics from the host and then some phenotyping information as well, and then sort of compiled that information to explore the phenomenon of can we change the feeding habits of farmed animals and does that change the sort of outcomes. And the digital twinning project from Rothamsted here is looking at things like soil microbiome data, crop yield information, including some quite broad scale sort of imaging techniques from drones, this sort of stuff. And then environmental data as well, and then combining that together in a way that you can start to build a digital twin, and you can ask these kind of what if scenarios. What if the temperature goes up by 5 degrees in the next 10 years? What's that going to do to crop yields? What's the likely impact based on what we know about soil microbiomes and the impact of temperature on those? So this is quite broad, quite coarse-grained sort of multimodal data integration. So there's sort of 3 different examples. They all sort of have a common thread, I think, which is how do you start to combine, how do you link those kind of different types of data? You can do that at different levels of granularity. One way that I think we've looked at in the past, in the previous life, I used to run the biosamples database at EMBL EBI. We used to spend a lot of time linking samples together. So, for example, you can combine genomics, transcriptomics, proteomics data on the basis of having a shared sample provenance. So whether that's an individual sample that you've done multiple different assays on in the first example, or you've got multiple samples derived from the same source that you've done multiple different experiments on in the second example, or whether you've just got sample metadata that allows you to link at some sort of geospatial kind of dimension. If you've got the same the geographic coordinates, for example, you can start to combine soil microbiome with imaging data from drones. So thinking about how you start to link those things together, I think, is sort of the common thread. So the kind of hook here is how do you link? And I think I wanted to finish with this slide, which is the sort of biocommons that BioFair is thinking about assembling. What I've talked about a lot is the bottom 2 sort of tiers on this pyramid, thinking about the sort of data commons and the methods commons. How do you annotate data consistently? How do you use shared identifiers in the data to link biosamples together? What methods could you use? So experimental techniques, can you align on experimental techniques to sort of do that kind of multimodal data integration? But I really kind of think that the main challenge is the one at the top here, which is the sort of people perspective. What we find, I think, in sort of our scoping analysis and in the work that I've done previously, is that actually the sort of methods, the biosamples database to do the linking, the methods to do the analysis and build a digital twin are there and exist. The challenge is doing the culture change, rallying the people around the usage of shared identifiers early enough in the process so that it's suitable and amenable to linking data on. And that was my last slide. So, and I'm just out of time. Thank you, Jeff. So, yeah, I will stop there.
Thank you so much. Do add your questions to the Mentimeter. Bernie, please take the stage. Local from Australian Biocommons.
Great. Thanks for the invitation, Wolmar and Jeff, to speak today. So I'm going to talk about a case study in multimodal data collection and ultimately integration. It's called, the PX for Health Initiative, and the PX4 stands for Predictive, Preventative, Personalised and Participatory. So what we're doing here is it's a collaboration with the University of Melbourne and Australian Biocommons and other partners to build an accredited platform that has really two main parts. The first part is what we call a phenotyping clinic or deep phenotyping clinic, where study participants attend in person a clinic. It's about 4 hours. They have a variety of tests done, for example, body mass index, DEXA scans, retinal scans. They answer questionnaires about lifestyle, mental health questions. They have a diet app where they do this prior to attending the clinic, where they follow their diet. Lots of things like that. They also give tissue samples, particularly blood samples. And the tissue samples are biobanked. And then that relates to the second part, which is a multi-omics platform for assaying those tissue samples in a standard accredited way. And the platforms are, there's three mass spec based omics, proteomics, metabolomics and lipidomics, and a fourth which is genome sequencing of the blood. And we collect all of that data together about each participant in the study, and it goes into a data commons. And we're using the Gen3 platform to put that data together as a data commons. You might have heard us speaking about Gen3 at various times during this meeting. It's a software platform, open source, provided by the Center for Translational Data Science in the University of Chicago. And we use Gen3 for a number of our data commons projects that we're working on. We've run a pilot study already for PX for Health. We've had about 160 volunteers go through the phenotyping part of the platform, and we've collected their tissue samples and they're being sequenced at the moment. So ultimately the view is to put this into production and continue onwards to larger scale. So one of the challenges that projects like this have faced in the past is that the data is often assayed at different locations in different machines at different times and then aggregated together. And that causes challenges, particularly for batch effects in the data sets. And if you've ever had to analyze this kind of data, you spend a lot of your life dealing with batch effects. And so that's a really big concern. And so the design of this platform is really directed towards avoiding those batch effects by having dedicated mass spec equipment. So that we've bought 3 new mass spec machines to do the multi-omics analysis for mass spec, and they're only used for this project. They're not research machines that others can reconfigure for other purposes. Obviously that reconfiguring of the machines causes challenges each time you set the machine up again. And measurements of those dynamic molecules like proteomics, lipidomics and metabolomics are very sensitive to the settings of the machines. That causes challenges if you don't have dedicated equipment for it. So I've already talked about this here, so I won't labour on this particular slide. But as I said, we've got 3 dedicated machines for proteins, metabolites, and lipids. These assays are all run from the same tissue sample. So I guess this is a assay-based multimodal using Tony's terminology, which I quite like. So thanks, Tony, for that. And then all of this goes into the data commons, as I said, which we're building at the moment. So one of the key things of this project is to establish a reference cohort of people from the community that are recruited through various other means to try and provide a control and a background reference for the data. And the other idea is, in addition to the reference cohort, the platform will be open to studies. So a research study can bring, say, a cardiovascular study in, and the individuals in the cardiovascular study will go through exactly the same processing that the reference cohort will. So we'll be able to compare the study against the reference cohort. This is leveraging national capability supported by Bioplatforms Australia all around the country. I won't go on to explain all of those because I don't have time. And as I said, there's additionally genome sequencing, which will be done by the Australian Genome Reference Facility, which is AGRF, and metabolomics is done through University of Melbourne at Bio21 in Malcolm McConville's group, and Australian Biocommons is doing the bioinformatics and data management. So what is multimodal data in this context? In my opinion, it's the combination of data from multiple modalities about the same individual. I'm nearly done. And about the same individual, and also groups of individuals as well. So we're interested in what's happening in individuals, but we're also interested more generally in the group. So I mentioned all the omics that we have, plus deep phenotyping, and the potential to link to population health and medical records, which is an ambition of this project. There is potential to include imaging modalities in studies. We won't be doing any specific imaging for the population or the reference cohort, although actually there is imaging inside some of the machines that measure things, including genome sequencing. There is actually imaging inside those machines. It's just buried, and the data you get back, it doesn't have pixels in it anymore, but it is there. So what are the challenges we're facing? There are many common issues. I've already talked about batch effects, and batch effects are, if you've ever spent any time analysing this data, the bane of everyone's life. It's an issue for reproducibility in science. We have this reproducibility crisis. People cannot reproduce experiments from others. One of the reasons is that there are strong batch effects in the data, which often strongly correlated with the outcomes that you're interested in studying. And this is especially a problem in longitudinal data. We're planning to follow individuals in this platform over many years and re-assay them over a period of time. Therefore, we can't mix cases and controls in the same experiment to try and avoid some of the batch effects that you might get through correlating data with your experiment. So we need to deal with that over time, which is an interesting challenge. There's the curse of dimensionality, which means that if you think of our data as a gigantic spreadsheet, where the rows are individual participants and the columns are the features we're measuring, we have vastly many columns, hundreds of thousands potentially of columns, and the rows are in the thousands. And that's not the dimension that you want to have for a lot of analysis. You want it the other way around. And so you can find a lot of spurious associations in your data when you have this type of data. So actually dimension reduction is going to be very important. Obviously machine learning and AI have a lot to say about that. Interpretability of the results is really critical, and especially if we're using advanced methods in AI and machine learning about what does it actually mean. And interoperability with external studies. So one of the missions for PX4Health is to be compatible with some of the major programs going around in the world. UK Biobank, for example, PRECISE in Singapore, and those we want to be able to combine and analyse our data together. So obviously we need to make sure we're thinking about those external studies and not doing things that are incompatible. Some specific issues, I think, so those issues that I mentioned above, the common issues are across the board. You see them everywhere when you're doing multimodal analysis. Some specific issues for this particular platform are that changes in technology, so that because we're planning to do this over many years, up to 25 years or more, the technologies that are used to assay these individuals will change for sure. Like that, there'll be different mass spec machines, different genomics technologies and so on. And we need to calibrate the data over time. I mentioned this yesterday, re-identifiability of participants and against privacy and consent is a particularly important issue, especially with AI and machine learning, and especially where the data is combined with other data sets internationally. And the last one is really a real key one, which is lack of methodological development in combining multimodal data. There's lots of ideas out there. At the moment, there's no obvious solution for taking this mass spec data and the genomics data and all of the phenotyping data and combining it into a combined analysis that has interpretable results. We're obviously working on that, but it's an open research question. So those are the challenges that we're facing. We're only just started this project. It's in its first year. It received funding through the Medical Research Futures Fund. So hopefully some of these challenges will be resolved in the time that the project runs, and it continues further beyond that. And I've mentioned this. I won't go over this slide because I've run out of time. But this is things I've mentioned already mitigate some of the issues, particularly around controlling for batch effects. Okay, so that's me. Thanks.
Thank you so much. Next is Maria, and she's from Germany, Euro-BioImaging. And remember, you can also vote on questions, even if you don't have your own. You can peruse and see what everyone else is asking and just top vote the question that's most interesting to you.
I've been said to be from Germany, but as you will hear, I am not from Germany. I just live in Germany, so I want to make that disclosed now. So I'm here to speak about foundingGIDE, and it's a mouthful, but sometimes I feel better when I hear about other projects like PX4. You know, the acronyms are always like a headache. So foundingGIDE, founding a global image data ecosystem. And foundingGIDE is a European-funded initiative, but we are global. We have partners in Australia, hence why we are here, also in Europe and in Japan. These are research infrastructures all over the globe. Our aim is to increase the coordination among image data resources, and we have a lifetime for now, about 2.5 years. We started in 2024, and we aim to include the community as much as possible in all our developments. Well, why foundingGIDE? Imaging is having a few challenges, and some of them this is not like a very complete list, but some of them are the image file size. As technologies keep developing, the individual size of every image is increasing, and each image contains, of course, metadata about the conditions of how the image was taken. Then, depending on the vendors of these machines, we have many file formats, over 160. And probably like the most disturbing part is that all the standards that are used to capture data about how the image experiment has been conducted is very fragmented, and hence foundingGIDE. What we are aiming in foundingGIDE is to bring together bioimaging and preclinical image data repositories and see if we can find some common ground. Our partners are from Japan, SSBD database, Bioimage Archive, which is hosted at EBI in UK, and IDR, which is developed by Open Microscopy Environment and is also hosted at EBI. And then on the preclinical side, our partners are from Australian Imaging Services and from Italy, the CNR, and these are for the preclinical side. But today, I will focus on the bioimaging data repositories in foundingGIDE. which are the SSBT, Bioimage Archive, and IDR. And what we attempted to do in foundingGIDE is to see if 3 bioimage data repositories can agree on a set of shared information that is enough to develop some interoperability, and then prove this interoperability by building an API. And on top of this API, of course, you can develop other applications. And just as a starting point, each of these platforms, of course, have their own data model that is already established. And also, like mentioned, would be that the scope of foundingGIDE is not to modify the data format of the repositories, but more find a common ground. And the starting point was that some of them had over 100-something items that they would save in the data model. Some would be required and some would be optional. And obviously, they are different. So the starting point is a discussion, a very, very long discussion. And what has been done in a very practical way was just to compare the data models between the 3 repositories and then come up with the shared set of information that is enough to describe images, and they are also compatible with the 3 very different platforms. Then having just the metadata schema is not enough. You have to have also ontologies to describe these schemas, to describe the items in the metadata, and this comes with other challenges, such as some of the ontologies used in the domain, they are not updated anymore, and then you have all kinds of challenges that you have to navigate around. And related to these 2 outputs, we already have the deliverables. They are all uploaded on Zenodo, and the next steps in the project would be to come up with a recommendation on how you can package this metadata, on top of which you can create APIs and create applications on top of it. And these are undergoing. They would be probably discussed at the next event, which I will speak about. And now maybe zooming out a bit and going back to the, maybe like the topic of this session, what we are doing in foundingGIDE is not focusing on the multimodal aspect of integrating imaging with other modalities, but we are creating the basis that is maybe flexible enough to allow any platform to integrate this sort of link to other modalities. And one of the examples that we are having in imaging, and this is implemented in one of the image data repositories, so that would be the Bioimage Archive, for the same study, which was looking at kidney fibrosis in mesenchymal cells. So they were looking for biomarkers of, I think it was fibrosis, yes, it was the biomarkers for fibrosis. They integrated the data from Array Express, which is transcriptomics, proteomics, and also the data analysis code was uploaded to GitHub, and you can have that all in a metadata specific field in Bioimage Archive. So this is possible, and this is also something that can be expanded on top of the recommended set of metadata items of foundingGIDE. But this is one of the main project partners, Josh, and he's been in this game for a long time. And one of the points he always brings is that creating metadata representation is easy, and the hard part is building consensus. And to this end, of course, we all know we have to bring the community together, hence today's meeting. And shameless advertising coming up. What we have also, one of the big focuses of GIDE is not just creating the technical recommendations, but also bringing together the community. And to this end, we are organizing community events, global community events. We had the last one in Australia, in Japan. And now we are in Australia. And tomorrow we have our second community event where we will discuss more about the technical developments in GIDE. And you are obviously all invited to participate. And if you can't, there's going to be one in Europe next year. And we have Germany quite represented here. So we would welcome you all to participate in the discussion on how we can create the best foundations on top of which we can link imaging resources and then imaging to other modalities. And I think I saved time like, for everyone else.
Thank you so much. And last speaker, Aastha, please join me on stage. Also from the same organization, but distributed across Europe, maybe not in Germany.
I mean, that's not right. Okay, hello everybody. Pleasure to be here. Already heard lots of nice talks in a lot of detail. So I'm just going to zoom out to a level where things start to become vague, but then still hope that it's useful, right? Okay, so let's start and see where we go with this. I am based in Germany. I do work for Euro-BioImaging, but today I'm going to talk to you about our efforts towards the European Open Science Cloud. And so what we are working towards is a life science node in the European Open Science Cloud. And this is not just me, but I'm working together with other research infrastructures, European research infrastructures, and working closely with Peter McCullum, David from ELIXIR, David Marshall from Instruct-ERIC, and Stéphane Pesan from EMBL. All right, so let's start with a little bit of introduction. Well, what is the vision here? So for the European Open Science Cloud, what it wants to be, or what the big vision there is, to provide us with a web of fair data and services. And this is as wide as it sounds like, and it is pretty much on the European level, right? Although they have intentions to be globally approachable and interoperable. So what we want to have is a trusted virtual environment that can support open science based on certain core horizontal services. This would basically make e-infrastructures and services more accessible for researchers across disciplines and, well, through Europe. From a researcher point of view, the idea is that you will have a place to store, share, process, analyze, work with your data, with your fair research outputs, well, trying to really dissolve the disciplinary boundaries here. Okay, where do we fit in? So basically this becomes then the EOSC for us. We want to be able to share digital infrastructure. We want to share data, software, practices, and knowledge in an interoperable way. And also, quite importantly, this also helps us work towards European policy to allow such developments and developments of such infrastructure and practices. Okay, are we there yet? No, we're not, but we are working towards it. And one of the ways in which we are doing it is what I'm going to tell you today about. So what we are currently focusing on is trying to establish an EOSC federation. So this federation is composed of various nodes, and these nodes can be, well, different in nature. So if you look at the ones which are kind of on their way of becoming a candidate node right now, you have things like, so ESR, so these are like photon and neutron, you have blue cloud, so again, more thematic nodes, as well as some of them are more national nodes as well. So we're really trying to group together these services at different levels that are meaningful and useful, and then find a way of then to interoperate with each other. Yeah, so there are many geographical communities as well, but we here form the thematic community. These are all the 4 region infrastructures here at the bottom. And so what do we want to achieve together? Well, in this wider picture of having a common data space for all of the fair data and fair research that is being done in Europe, we want to form a life science dedicated node. This would be a dedicated space for life science research to discover and access data that is data and services that are useful for them. And with that, we want to be able to achieve, let's say, a place which is more meaningful from a researcher point of view, maybe with a higher level of curation, with a higher level of interoperability, and kind of going in the direction of enabling the multimodality that we've been talking about at different levels. Like I said, the partners include, and what puts us in a very interesting position here is that the partners that are coming forward in this, initially in the first step for this formation of this node, include Euro-BioImaging and Instruct-ERIC and EMBL, which actually go all the way from producing the data, so really working with the researchers and trying to make sure that the fair practices can actually be adopted at that moment, right? So we're going all the way from instruments, like data acquisition there, and then trying to make it available on a larger scale, and we are working together with data infrastructures like ELIXIR here. The diversity of data, yeah, so it's fairly wide. We have, of course, things like structural biology data with all the different modalities like X-ray imaging, cryo-EM, NMR, and so on. We have imaging data, which comes all from microscopy technologies as well as biomedical. More going into, we heard a lot of examples of biomolecular and multi-omics data, as well as more correlative data. So again, now I feel like I should be using Tony's terminology in future. Very happy about that. So really various kinds of data sets, whether they're either produced together or separately, but really trying to make sure that there are ways in which we can work together with this data. So there's enough layer of interoperability between these data sets so that we can actually enable some sort of secondary reuse of this data. All right, so of course there are various kind of services that these nodes bring in. These research infrastructures bring in. I'm not going into a lot of detail here because this is rather technical, but we will have services like maybe more consultation services, so more user-based, more user-focused, things which actually go all the way from instrument data acquisition, trying to make sure that metadata is right, all the way to deposition services. There are various core data resources that ELIXIR brings in, and from EMBL there's also a lot of focus on multi-modal data and a lot of infrastructure databases that they bring in. So it's going to be rich, is what I can say. I want to leave you with a very practical example because when I want to think about, well, we did talk a little bit about what level of multimodality, right? So one of the examples of what we think a space like EOSC is going to enable for us is basically staying not just within our domain, which is life sciences, which is something that we want to bring to our users, but also the solutions that we have. We want to bring them to a wider community. And this project here, which is also funded by the European Union, called the OSCARS project, is one of the open cause projects called OSCARS Fiesta, which is coordinated by my colleague Beatriz Serrano Solano. It is a very nice example of showing how we can actually do that. So, well, with this project, we could bring together communities that have similar needs towards image analysis. We had FAIR-enabled resources that actually allowed us to pull together data, in this case, imaging data, and, well, not just bioimaging, right? So here we can see some nice cells. Well, this is astronomy data, and this is environmental data of some tree canopies, right? And have common workflows, or like show the power of the common workflows. So we did what's called a Voronoi segmentation to allow us to, let's say, well, count cells, galaxies, or trees, right? So, so if we actually have this sort of a place where we can combine all these data, we can take advantages of methods that might be developed in one community and, well, see how far we get with it. Well, with that, I'm going to stop and hand it back to Wolmar.
All right. Thank you so much. All right, so we're going to head into a Q&A session, and I would like to welcome all the speakers back on stage, so I should have warned you about that. But maybe you want to pick something up. You can't see the screen, so maybe if you have a phone or something where you can see the questions or what's being presented, then you can bring it with you and just sit yourself here while we go through a few questions. While we're stepping up, I'm going to switch over to the Mentimeter. And I'm going to see if I can get, there we go, I can select the questions. So this is one of the top voted ones, and I'm going to try to pick one for each of you. I'm going to read it up, so hopefully you can remember and just ask or answer the question as you will. So any reflections on what the common focal points for integration are, e.g. Molecule, cell, tissue, organism, population, or something on those lines, and then there's examples, UniProt, biosamples, where is the global consensus? Tony, that was during your presentation.
Yeah, we got microphones.
Oh, very important detail.
Thank you. Okay, this is on right. Yeah, there is. I don't think that there's not a consensus around this, and I spent a number of years leading the biosamples database, and I've had this conversation very, very, very many times. I think ultimately it boils down to a trade-off, right? You can about how deep you want to model. So you can either get really, really, really complicated and model very explicitly sort of all of these things down to very fine levels of granularity, and then you have a very rich approach to linking things together, but it becomes incredibly difficult to actually tease through what you've done afterwards. You end up with a really, really complicated knowledge graph, effectively, that you have to try and rationalize, versus actually modeling something quite simply so that it's much easier to integrate, but you lose some of the expressivity. What we've found over a number of years is that it's very use case dependent. So the only advice here really is think about the sort of anticipated reuse of the data that you're actually aiming for, and work backwards from there, rather than sort of thinking about exactly how you've collected the data and modeling that perfectly. So to give that's a very general answer. To give a very specific one, we had a number of conversations with people that were doing plant crossbreeding and had a really complicated crossbreeding program that resulted in specific sort of plant strains being developed and grown, and they had a seed store for these things with 20 generations of crossbreeding that had happened previously. And you think, should you model all that out? Actually, you probably don't need all of that, as long as you've got a sort of biosamples identifier for the last one and some sort of passport information about how that sample was created. You've at least got enough information to go tease through that if you want to. You don't. It doesn't become free of work, but you have the ability to do it rather than not. So I think that's a non-answer to the question. There's no consensus, and it's actually pretty hard to figure out sensible guidelines in this space. But the recommendation, I suppose, is start from what you hope to be able to achieve at the end, and then work backwards from there.
All right, thank you so much. I hope that answers the question. Is there anyone who wants to counter that and say that yes, there is, or just want to nod or stamp off that this is a challenge?
I mean, I can add by saying that, it also depends on how the data was acquired. If it's data that you're acquiring for big atlas projects where you are going to acquire different kinds of data, you have standard operating procedures and so on, yes. But, if you just want existing databases to have interoperability there, that is going to be fairly limited and fairly superficial. So I think we can make use of these places where we have, let's say, a deeper interoperability possible. But I think there is probably some utility in trying to do this at a superficial or shallower level, not superficial, as well, because just to make sure that the data is a bit more integratable.
All right, thank you so much. So marking this as answered, and the next one is for Bernie. Using dedicated mass spec instruments does not sound very scalable. Do you consider implementing some calibration approach?
Okay, good question. Let me think about it. I mean, the machines are calibrated, of course. I'm not sure why the questioner is saying it's not scalable. I mean, this is for this platform. One of the things that has changed recently is that new high throughput mass spec machines are available, and that's what we're using now. It's kind of revolutionizing mass spec in terms of turnaround time and number of analytes that you can measure. So the other thing, I'm not really answering this question directly because I'm not sure I understand exactly where it's coming from, but the possibility to do absolute quantification now is a new feature of these platforms where you're actually trying to measure the real quantity rather than a relative quantity. And that's a big game changer for these kind of measurements, where the problem with relative quantities is, again, I banged on about batch effects before, but that can't stress how significant batch effects are in scientific data. And if you can do absolute quantification, then you can mitigate that a fair bit, because you're actually trying to measure. And then if you change the platform, assuming you can do that again, you've got a better chance of removing some of those fairly serious batch effects. Anyone who's done RNA sequencing or DNA methylation knows that you spend almost all of your life normalizing the data and trying your best not to throw out biological information at the same time. So I'm not really addressing the scalability question. I mean, Pigs for Health is it's got its own scaling limits in terms of the number of participants, but actually the mass spec machines are not the scaling limiter. The scaling limiter is the dollars to do all of this work. It's very expensive. So the number of people we can get through is in the thousands, which is plenty for what we're aiming to do. I'm not a mass spec person. I have to put my hand up and say I'm a genomics person. But what I understand from the other experts in the team is that prior experience with using platforms from different providers at different times around the country and internationally has really created data sets that are not comparable because of these variations. And so that's the reason why we've got dedicated hardware for it. It is expensive, but it's also trying to move mass spec and analytics from a very research-oriented platform, which is great for, into a clinical platform, which is really exciting, because genomics only gets you so far. And I say that as a genomics person, it really gets you to a certain point, but really to understand the dynamic behavior of cells and individuals, then these protein-based systems are really powerful. So that's why we're pushing this into an accredited platform for that reason. So I don't think I really answered the question. I'm not sure I entirely agree that it's not scalable, but yeah.
Yeah, so thank you for that answer. I mean, I realized that maybe the focus also on the panel is a little bit maybe more on the integration points. And I mean, I think you addressed, I mean, some technologies are a little bit more mature than others. And I mean, where you can really aim for the integration will depend on maybe on the technology itself, how established it is in industry as opposed to I mean, an academic actively developing setting and things like that. Anyone want to expand on the same question, or should we take the next one? All right. So next question was also for Bernie. So maybe I'll jump over that one, and we can go back to it, so everyone gets a question to start with. So Maria, there's a question for you. So are the descriptive models for the GIDE global image data ecosystem aligned with those of omics and other modalities, e.g. for samples, molecules, cells?
So I will define first what I understand by these descriptive models. So what foundingGIDE tried to define was first the metadata schema, and then for each of the items in the schema, or the recommendation harmonization metadata structure, each of these items then has an ontology defined. So each time you have information in this schema, you can refer to an existing ontology. And the way we intend to package this information in the raw crate package gives so much flexibility that you can create whatever bridge and cross-talk with any other ontology from any other domain. So what our approach was first of all, have a set that describes the images enough, then make this relevant for the everyday use. And having that discussion starting point, we got in contact with many members of the community, and then we had feedback on if the things are actually helpful in reality, and are they solving any of the community current problems. And this was an iterative process and is still ongoing, and it will probably still be developed as long as we need metadata schemas. So the answer is a yes, and if it's not a yes today, it can be worked into the solution. So we tried to be aware of the future needs of the metadata schema, and I think the way we would package the solution, if that is a foundation that is flexible enough, then the answer would be yes, we did.
Fantastic. Anyone want to expand on the same question?
And maybe I can expand the question to the other maybe speakers. How do you see the schemas that are perhaps the one that you use with your cross modalities? How do you solve the interoperability at the metadata level?
I can have a go at this. So I noticed there's another question that's for me that's sort of in this vein as well, which is a little bit about how is multimodal integration possible, and are individual repositories too specialized for one purpose? And in Biofair, we're trying to encourage people to think a little bit more about data journeys rather than data repositories, because I think what we do in the life sciences domain quite often is think about a data repository, and then that data repository has to have a set of metadata standards or a schema, and that should be mapped to all the other data repositories that are out there. And that's a very siloed way of thinking about the world, right? So, and you end up with specialist data repositories, which are really, really good for one thing, but then become incredibly painful to break out of that silo. And rather than thinking in sort of, okay, I've got a database and I need people to put data into my database in my standard, and then other people can read data out of my database using that same standard. Instead, thinking about the world as a set of transformations along a journey that goes from assay to insight, and then thinking about the hooks that you need, the bridge points that you need to be able to connect those transformations. And very soon you end up in the, well, actually the thing that's most important is persistent identifiers and good provenance. So rather than necessarily thinking we need one standard to build them all, there's 3 different databases. Wouldn't it be great if they all had the same standard? I'll build a new database. Now you've got 4 databases. The XKCD sketch that I'm sure everyone in this room is totally familiar with, right? And rather than thinking in these terms, to think about, actually, it is totally fine that you can build a database for a specialized purpose, and then the next step along the journey is to read data out, figure out how to link it together, and then sort of define your transformation in a sort of robust way. So I think, that's part of the reason why I advocate for things like biosamples identifiers. It creates those kind of hooks, those bridge points, those things that you can link to. And thinking about the kind of use cases, what likely journeys do you need to support from assay to insight, then encourages the community, I think, to think a little bit broader than just what's the standard that I have in place for my database. It's a culture change problem though, right? That is.
But maybe I will just want to clarify, not that you insinuated that, but in case I wasn't clear enough, we are not creating the 4th database because that would be a pain. We are just exporting the metadata, so the data still sits in the same place. It's just the metadata that would get exported to a store, and then from that one you index it into an API and build the application on top of it. So we don't want to be part of the meme, or that would be.
All right, so thank you so much. I'm going to bring up a question for Aastha as well. We have, what obstacles do you expect concerning semantic data interoperability within and between EOSC nodes? So I guess this is cross-disciplinary, then also cross-infrastructure related question for those who don't know or remember EOSC. Will EOSC favor few centralized repositories to reduce the complexities of interop?
The answer to that is, it's easier to just say no, mostly because I don't think EOSC wants to, the idea is not that, oh, stop doing what you're doing, but actually follow what we say. That's not the idea. I think what they want to do is that they want to preserve the richness of all the things and all the methodologies that people use. And still be able to still make sure that there is some sort of a common communication, and that's why any kind of integration there is at a very shallow level. So I wouldn't say, I know that the second part of it was around if there's going to be favored with some centralized repositories, but I mean, yeah, obstacles. Yeah, pretty much the main obstacle here is not just that it is different kind of data. It's not even just life sciences data, right? It's all data, all research data that is being produced. So there is, I mean, it can only remain meaningful if, you know, that's one of the reasons why we have different nodes, so that you can have a little bit more level of description, a little bit more specific utility for people who are coming from a particular domain. There are also other interesting examples like national nodes, but that's more, you know, in case you want to use some national infrastructures, that's where you can get your ability to get your compute. But if you want to use the national compute and actually deploy a life science service, that should be made possible, and that is the interoperability that EOSC would want to enable. So challenges, I mean, there's no lack of challenges there. A lot of them just end up being very political and legal, frankly. Lots of talking and lots of paperwork. But yeah, I mean, we're just hoping that, you know, everybody has this bigger idea of working towards that interoperability. And that's why we are starting to do this on the life science level, at least from our point of view first, to be able to serve our users in a more meaningful way and then interact with a larger landscape.
Fantastic. So can I paraphrase, and maybe you can correct me if I'm wrong? So I'm thinking what you're saying is basically you can always go to the individual infrastructures that are partnering in this and get services directly, but at the top layer there should be some sort of entry point for those who might not really know where to go, or if you want to do some cross-disciplinary work that hasn't already been kind of well established. So I mean, do what we still are doing well, but find some way of having common ground where there's no direct conflict, like compute there. I mean, maybe people are doing that more or less in the same way.
Yeah, and also it kind of gives us an excuse to work towards more common ways of working. So like just the different research infrastructures that are part of it now, we are now talking to each other. We intend to talk to more life science research infrastructures over time. So yeah, I think it's I like to think that in case of EOSC, it's not necessarily the destination that is so charming, but it's the journey that is going to, you know, yield something interesting for us.
All right, any other reflections on, I mean, maybe not specifically EOSC, but I mean, fitting into a larger ecosystem and integrating your data resources or services into a broader context?
Yeah, I could perhaps have a go at this one. So, you know, I sort of challenge the premise a little bit here that more bigger centralized repositories reduces the challenge of interoperability. So, on the subject of interoperability, I think I am an outgoing ELIXIR interoperability platform lead. And so when we think about interoperability, quite often we think about, rather than thinking about it as a function of a repository, so again, in the spirit of data journeys rather than data repositories, interoperability is really a sort of contract between how the data was generated, the people who are generating the data, and the people who are doing something with that data, that downstream usage analysis, whatever, deriving insight from it. Now, when you think about interoperability in terms of reuse, that essentially says, well, I want to reuse the data for something that wasn't the purpose for which that data was originally collected, which means that you've got a different contract, right? If you have a contract in mind that says, I'm collecting the data for this purpose, you've got an implicit agreement between how the data is being generated and how it's going to be used. Now, if you bake that agreement into a single data repository, you're never going to be able to reuse it for any different purpose. So that's your challenge, right? It doesn't actually matter if you've got multiple repositories there. If it's multiple repositories, but you've still got the same agreement between how the data is being produced and how it's being used, you've still got the same problem. And to some extent, it's actually easier to say, well, we'll collect the data once and then use it for lots of different purposes. So we'll build up an ecosystem of repositories that are focused on the particular question we want to interrogate that data for. So really, I think the sort of the insight, I guess, that I could offer here is that if you think about data journeys rather than, you know, one standard baked into a single repository that solves everyone's use cases, then your life becomes a little bit easier. So, you know, if you have one centralized repository, you have to sort of centralize all the requirements gathering for how all that data is going to be used. And in my experience, that's always a bottleneck.
All right, thank you. So, Bernie, is it okay if I put you a little bit on the spot so we get a non-European perspective on the same thing?
Sure.
And maybe we can also start moving the chairs a little bit, because I realize for the next part, you might want to be able to see the screen. So if we just shift, I mean, a little bit horizontally.
Sit here.
All right, so some reflections on how your infrastructure fits into the larger picture in Australia and perhaps globally, and if, I mean, something fits the same general line of inquiry.
Okay, so I'll keep my response in the context of the PX for Health program, even though there's lots of other things we're doing in Australian Biocommons that would be relevant. So for PX for Health, I mentioned that one of the ambitions was to have the data be analysed in the context of other international multi-omics collections, such as the UK Biobank, PRECISE in Singapore. There's also a number of other commercial kind of organisations developing these. I won't mention them by name because I don't want to necessarily promote any specific thing. But there are those happening. Actually, there's many more of those than there are necessarily research ones as well. So I agree with Tony about the research journey too. I think that's a good perspective. I mean, what I can really say is we have spent enormous amounts of time investigating what is done in the other programs and trying to align as best as possible everything we're doing with those. I mean, the challenge is that some of those, like UK Biobank and so on, have made decisions that were decided decades ago. And you don't necessarily want to be tied to decades-old decisions. And that's not criticising any of the work. That's just the reality that those things happened a while ago, and new technologies and things are available now. But lots of time has been spent on thinking about making it future-proof and being compatible with other data sets as well, in terms of technologies used for collecting the data, the way the data is represented. And yeah, so I think we've put in a lot of effort to make that possible. We're thinking of this being a program, if it's successful and we can keep going and get funding, we'd like it to run for 25 years or more. So where it's a long-term program, you have to think about what decisions you make now, how do they impact the future in a long term? And that includes just standardizing on workflows and things that we don't want to lock in bad decisions now that would affect things down the track. So that's a wishy-washy answer, I suppose. But I guess the best we're doing is trying to make sure that we're very well aware of what everyone else is doing and trying to also say what we're doing and keep them aligned. Yeah. So I mean, we're so early in the process; it's hard to say how that's going to turn out. Yeah.
All right, thank you so much. As you might have seen, I switched over the Mentimeter to a new slide here. And I mean, I would like to hear the perspective of the audience as well. My idea here is that we will continue the discussion. If you don't have any comments, I will bring up a question from the Q&A and see the discussion. Otherwise, I would like to kind of give you the opportunity, those of you sitting in the room, to maybe more than a question, give a provocative, this is something that I think we should be doing, or something that I would like to be working on or connecting on. So you can type into the Mentimeter if you don't want to speak out, and for those online, you can also use the Mentimeter. I do see some hands, so...
that microphone running.
You're the runner as
so
well.
I can avoid answering.
Good idea. I was I think it's maybe a Tony question, but I don't really mind. I was thinking how you had the final triangle figure with the people and thinking about culture change. And I've done a little bit of, engagement, and I noticed that mostly the people that engage when it comes to things like metadata or linkages and working through that are often employed in that role, not necessarily researchers that, that's their bread and butter, but they sort of, it becomes an afterthought and they don't have time for that. So I wondered if data stewardship, in the sense that we really facilitate that as the avenue to make sure the data is fairly available and interoperable, is the way to go rather than relying on culture change. Otherwise, I thought, what's your ideas for culture change? But that's also a hard question.
Yeah, I can totally take that one, and like, amen is the answer. I think is the brief answer. So, I mean, I think I would say my perspective on culture change here is mostly that we need to build bridges between researchers and good data stewards, so people who are sort of positioned in this slightly more technical role to support research. So and get sort of expanded kind of responsibility and leadership in those kind of research technical professional or research services or data stewardship type of roles. So these are the people who really care about kind of making sure that it's done right, when you're collecting metadata, that it's done right, and embedding those processes early in the data journey, So I tend to think, just to give an anecdote, right, from sort of my time at EBI, a long time ago, actually, it must be 10 years ago, we did a survey on people who submitted data to EMBL EBI repositories. And what we found was the vast majority of people who submitted data to EBI repositories did it once and then never did it again. There was a number of people, it's something like 90% of submitters to EBI repositories were first-time submitters. Then something like 9%, so taking you up to 99%, something like 9% of submitters were sort of power submitters or intermediate submitters that had done it kind of 8-9 times, something like that. Had done it a few times and had got some expertise. And then 1% were large-scale data brokers that did it hundreds and hundreds of times. Now, this is an old figure, and I think in that period of time, the role of data stewards and brokering services has grown quite a lot. But I would be very surprised if those numbers had changed significantly. And not only was it the fact that was the sort of profile of the people who submitted to EBI data repositories, if you looked at where the data came from, it was almost exactly the reverse, right? So the 1% of people submitted 90% of the data in terms of data volume. And I think that just points to the fact that there's actually sort of relatively low uptake within the community of data stewardship, because most of those people who submitted once are researchers who've written their manuscript and are told by the journal that they have to submit their data set to a public repository and go, I have no idea how to do this, and do clear the lowest possible bar. Whereas the other 10% are people who actually care about this stuff. So I think the culture change here that we need is to grow that proportion of people. I'm sure that it's changed in the intervening period of time, but I am confident that there's still a lot more to do.
Can I add something on that? I think I agree totally. But I mean, my experience is that funding agencies, at least in Australia, don't put funding towards data management. They don't even notice it or see it, and it's a cost. And I see a hand up there, so I'll come to that too. So, every chance we get, should be advocating for research data management in projects. The other thing is, it's what you measure is what gets optimized. And there's a lot of credit for getting your name on a publication in a journal and so on. There seems to be very little recognition in academia, from my point of view, about quality of data, and actually I think that's a cultural change too, is like if your data is being reused, you should get lots of credit for that. That should be a big win and seen as a success. It's not necessarily seen as a success at the moment, and I think if you have real incentives for people to do that rather than just saying you should do it, I think you have to do that too. Otherwise, people are busy and they can't. You know, academics are struggling to keep their careers going and things. So if we want that, then we need to. It's a big change, I think, really.
Yeah, I've got the mic. Just adding on top of it, Marek Cwecer, Herowski Institute, Prague. We have established two different levels of data stewards. One is technical, which is really trying to develop and help with the technical issues and the more complex systems. But we are very intensively working on having a data steward in every group, any scientific group. Someone who is already a part of this group, and we are lucky because in Czech Republic and in European Union, the data steward is eligible cost for the scientific proposals. On the other hand, it's taking off from your budget, so you can argue that this is not really extra money for data management. But what I wanted to say is that this cultural change, we already in 2 years observe, of course it's very slow, it's very individual, and that the knowledge is being transferred from data stewards to the researchers. The process is slow, but it is there. So if this runs, if there is a critical mass of data stewards, you can deliver this information slowly to the research community.
I have one comment, because I am more on interoperability level. And I have, from life science, I have experiences from 2022 in genomics data and on biobanks data. And also what I have seen, many different standards, many different vocabularies on metadata level or also on data model level. And this is not so easy to integrate. And also what I have seen, every European project is, how I say, is one island, and you produced one registry with one type of data sets with one metadata standards. But on other project, they produced the same things. And this integration, I don't know, I don't see vision in European Union on this level, how you will integrate things, because everyone wants to do as island, because in new project, you will have funds to do things further. And from my point of view, I am a practitioner, and I want to use your data sets. And for every registry, I need to know this metadata schema. I need to know data model of maybe every data set inside this registry. So this is critical problem, how we solve this. It is possible, for example, in genomic to use this G4GH standard on some level, but on data model level, we haven't agreement how to. Also, what is interesting, you have different data sets and different standard operating procedures. And it is not possible to compare things because you don't know how this data has been produced. For example, in bioMRI, they started with metadata schema for standard operating procedures. And after 1-3 months, they have seen that this is very complex process. And so this is my point of view on this integration. But all is possible, like Aastha said, if you have agreement and achieve agreement on the level of European Union, and it's, of course, it is possible to do on the level of the world. It will be very helpful for all of us.
All right, thank you so much. It's time to start wrapping up a little bit. I have tried to add a few interpretations of the questions with, I mean, my view as well. So I think there's a lot of things that we could work on together. And if you are really interested in working with, I mean, maybe me and some others who really would like to end up with a document that describes the situation in a way we can have informed discussions with our colleagues and maybe also with the global community around multimodal data integration, then please do share your contact information or some names of groups that we should try to engage with going forward. So I'm going to leave this one open, I mean, even after the session closes, so you can add some suggestions for groups to reach out to. It could be your own or it could be something else. And then there's also this shared notes document. If I scroll down here, I see that some of you have added your names, which is great. If I scroll down even further, we will see that there's already some notes here. And after this session, we're going to work on tidying up these notes, complementing them with maybe some information from the Mentimeter and the recording. And I will send an email to those of you who shared your contact information in this document. So maybe this could be a start for some collaborations. Feel free to also add additional links to relevant communities that we could work with. Hoping to see you engaging with the Research Data Alliance and our interest groups at future events. And the next one is sometime in March, so do check out the Research Data Alliance website and sign up to be a member if you're not already. It's free. With that, I think the content for the session is over, but the discussions will continue. We didn't answer all the questions. So if you had a person in mind that would answer your question, you can hunt them down during the breaks. I mean, it would be fantastic if we could find some future collaborations based on this. And in particular, if you know someone from the other regions of the world that are currently not represented in our interest group, please do help us get a contact so we could maybe broaden the discussions and make sure that we get a globally connected set of life science data infrastructures. So any last words from the speakers? Otherwise, I think it's time for some applause. So Barney.
Can I just make a radical, I mean, this is something that was on my mind for a long time about data management, data stewards and things. The situation we have in life sciences, I think, is a lot like in computing. You shouldn't get the programmers to write the test cases for your code. But that's what we're doing in the best case, in fact. And I actually think we should really encourage and have a culture of people trying to actually really test the quality of the data, and finding problems is not a problem to be hidden away. In fact, it's the opposite. And so I think actually we should be really thinking about data quality and interoperability and actually promoting work done to actually test this out rather than just assuming it's all okay. Because I think that's a real issue, and that should be part of a role. I think it shouldn't just be, you know, only thinking about the metadata representation of those things. It should be thinking about, well, there should be a position in the world which is saying, I investigate the quality of the data, and you know, I'm really trying to invalidate the data as hard as I can. That makes the best data that we can have. I think that would be a good thing to have.
I think that's a very nice, provocative end to the session. So, I mean, I agree that it would be really nice to explore new venues for, I mean, use cases and roles in making sure that we have good quality data. And, I mean, multimodal integration, if you want it to work across studies, then, I mean, that's a really big challenge if you don't have good, something that you could just trust that it's okay.
So we're going to finish on data quality at peer review of papers then. Data quality assessment, peer review of papers.
Yes.
Easy.
I think that's also
Easy,
a good
right?
idea. So, I mean, please do share ideas in the document. The Mentimeter is still up, so go ahead. And I forgot.
Yes, there's
sorry.
one
There's
final pitch that basically
one pitch here.
fits into this perfectly. So my name is Nils Hoffmann. I've been part of the MOMSI working group in RDA, led by Lindsay Anderson and Tim van den Bosche. And as one of the central outputs of this MOMSI working group, we have reviewed existing standards. You can go to the next slide, please. Throughout the life sciences world, basically. And we have created 2 deliverables already. So one is the landscape review that you can check out. And that's also now available as a standards collection in fair sharing. So that interconnects different existing standards across genomics, transcriptomics, imaging, and mass spectrometry. And basically also will serve as a basis for future activities. So I think that is a very good foundation to look into integration scenarios, basically, between multi-omics applications. And so something that is coming up now is going to go into this checklist template for gap analysis, and then also some final recommendations and an article potentially. So I would also like to invite you all to participate in this group if you're interested in working actively on standards and basically also promoting the collaboration in standard. And one last thing, I'm also involved in UPO-PSI, so the Human Proteome Organization's Proteomics Standards Initiative. On the metabolomics and lipidomics side in mass spectrometry, we're developing standard formats concretely and also engaging with the community of tool developers and repositories to basically, develop formats that are interoperable between the different participants in the ecosystem. And I think that's a really important part, and that's something that we might also want to continue in this group or also in the, in the other group, in the Interop group. Thanks.
All right. Thank you so much, Nils. An applaud. And I'm really sorry, this was a key part of the presentation originally planned. And I mean, it would be fantastic if some of you are working on similar initiatives, if you would like to share a slide or something else, you could just put a link into the shared collaborative notes document. So thank you so much, and let's continue the discussions offline. Thank you.